And welcome everyone for today's live session. So I hope my voice is audible to you. So today we are going to begin a new session or new series that is Sunday Journal Club. This is going to be a weekly series and so in this weekly series, we are going to discuss some recently published research articles and specifically related to bioinformatics, where we will be discussing the major findings of those researches being made, specifically related to bioinformatics, and what are the key aspects of those researches, and what are the research gaps which will lead to basically new uh, what you say, which can lead to further research in the field of bioinformatics. For today's session, I have chosen a specific paper which we have published, or rather, this is being published by me and my colleagues. And this paper was published in the journal Microbial Pathogenesis in July 2026. And herein we use QIIME2 for establishing an association between gut dysbiosis in diabetic nephropathy. For this purpose, we used publicly available 16S ribosomal rRNA data, and to be very specific, we used 108 samples across six projects or six studies where we compared the 16S ribosomal rRNA sequences across diabetic nephropathy and healthy controls, and the data was analyzed using an optimized and standardized QIIME2 workflow. So I basically choose this paper. So first of all, diabetic nephropathy is a major complication of chronic diabetes. As we all know, one among four Indians is suffering from diabetes as of today. So, and in most of the cases, it has been observed that with the time, the diabetes is going to progress to many other secondary complications or comorbidities, and diabetic nephropathy is among one of them. And going forward, it has been estimated in various researches and various regulatory organizations that diabetic nephropathy is going to be the chronic kidney disease is going to be the fifth most fatal disorders by 2040. So when we look into the diabetic nephropathy, there has been a growing evidences have suggested that gut microbiota dysbiosis has an association with renal inflammation and the progression of diabetic nephropathy. So there have been various individual studies have been conducted where microbiome data has been found to like a slightly going modification in animal models. Yeah, and this has been used or primers or sequencing depth, and for this purpose various primers, sequencing depth and bioinformatic workflows have been conducted across separate studies. Now, but there hasn't been many studies where a standardized workflow has been conducted across multiple studies. So, can an optimized and a standardized reanalysis of raw sequencing data has can it reveal new insights, or it can rather identify reproducible microbial signatures of diabetic nephropathy? That was our key question in this particular research. So, if we just look at this study briefly or the methodology of this study, so we basically conducted a meta-analysis or rather a pooled reanalysis as that was advised by one of the reviewers of this paper. So they asked us to basically modify this terminology, and accordingly we conducted a pooled reanalysis of publicly available 16S ribosomal RNA sequencing datasets. So across those six data sets, 108 murine samples were conducted. Of course, means these studies were conducted in mouse, And out of these 108 murine samples, 38 belong to diabetic nephropathy, 37 will belong to healthy controls, and 33 belong to diabetic related samples. Uh, One more thing which I would like to mention as a research gap in previously reported data is that in majority of the previous studies, what we have noticed earlier, that diabetic nephropathy is compared with only healthy controls. Means majority of the studies uh, they lacked another control of diabetes. So we basically thought of that we should include diabetes as a separate control to to see what is the difference between healthy controls to diabetes and to the diabetic nephropathy as the disease is progressing. So after screening, 6 independent bioprojects were included, and these bioprojects or these 6 datasets were included from publicly available repositories. So the key theme or the central strategy was basically we from NCBI we basically collected raw paired and fast queue files, and they were subjected to treatment through a single unified QIIME2 pipeline. So this was done to ensure that our analysis has a uniformity across all the samples. So once we identify that we wanted to like perform the analysis on mouse samples, so then we look out for we search for what kind of data sets can be available. So therefore we screened various publicly available repositories across NCBI, SRA, DDBJ, and ENA. And the inclusion criteria for these studies included that it should be murine samples, and specifically, we basically selected stool samples, and there should be amplicon sequences or 16S rRNA sequencing. And after the sequencing, we basically selected paired and FASTQ files and the relevant metadata. And out of the selected datasets, over total 6 number of datasets were retained. You can see all of them on your slide on your screen here as well. That is PRJ DB6437, then PRJ NA881044, PRJ NA817385, PRJ NA865044, 876080, and PRJ 1044925. So one important point which I would like to mention here is that the different 16S hypervariable regions were represented across all the studies. So once we basically for performing the bioinformatic analysis, we basically downloaded the SRA files from the NCBI and then converted into FASTQ files. Then we perform a quality control analysis using the FASTQ, and for multiple files, we perform multi-QC. Once we had the FASTQ files and quality control performed over them, Thermometric was conducted. Thermometric was applied to remove the adapters, and so that we can have relevant files have which basically meet the quality criterion uh, for downstream processing. After that, these FASTQ files were imported in QIIME2 using the manifest files. And once the manifest after this after the QIIME if you don't know that QIIME2 basically does not use these FASTQ files directly, it converts them into artifacts which are known as .qza files extensions. QZA stands for QIIME2 artifacts. So after that, these QIIME2 artifacts, they were subjected to DADA2 denoising for error correction, chimera removal, and generation of amplicon sequence variants. Once these ASVs or non-chimeric sequences are generated, they are basically aligned to the reference database, that is Silva database. And once they are compared with the Silva database, so then we basically conducted alpha diversity, beta diversity, and finally statistical analysis was conducted using ANCOM BC2 for analyzing the differential abundance across the different datasets. So, before moving forward, if you don't know how to basically download the SRA files, how to perform the FASTQ files, or how to perform the quality control analysis, and how to import QIIME2, all these things I have discussed in my previous videos, including up to alpha diversity. You can check them out on my channel as well, and I will also leave the link of all these videos into the description. Moving forward, so we basically conducted the fast quality control. For quality control, we performed both fast QC as well as multi QC. So all these indicated high quality reads, and duplicate reads. They are range from 92 to 98%, and which was consistent with the PCR based amplicon sequences. The mean GC content ranged from 48 to 57%, and after trimming, nearly 30,000 to 40,000 high quality reads per sample were obtained, which were basically sufficient for downstream profiling in QIIME2. Uh, an important point one must mention is that there can be a high duplication in amplicon data, which does not necessarily translate into that the same thing as duplication in a shotgun sequencing. So once we have basically performed the QIIME to import and converted the FASTQ files into the uh, QIIME to artifacts, they were subjected to DA2 denoising steps. So these denoising steps remove all the chimeric sequences, filter out those chimeric sequences, and retain only the non-chimeric reads, which are also called as amplicon sequence variants. So it was noticed that these non-chimeric reads or ASV retention, it varied very much substantially across the six data sets. The mean percentage of the reads that pass this filtration sets, they range from 32% to 98%, and non-chimeric reads, they range from 29% to 78%. So you can see that there is a lot of variation. However, we basically kept a threshold that reads below this 29%, they were not basically used for downstream processing. So despite variabilities, the sequencing depth was sufficient, and that was suitable for downstream ASV and diversity analysis. So one thing you may question is that does a common pipeline can fully remove the heterogeneity introduced before the sequencing, because all these studies have been conducted separately under separate uh, conditions. So obviously one can question the authenticity of this whole study, that whether there can be an inherent heterogeneity. Of course, from a scientific point of view, one cannot deny that there can be a heterogeneity uh, in the data sets, because all these studies have been conducted under different conditions, under different geographies as well. So but still the analysis parameters right from like quality fast q analysis or fast qc analysis up to alpha and beta diversity or ancom bc2 analysis, all the parameters were retained consistent across all the six data sets to minimize the heterogeneity that can be introduced before the sequencing. So we basically tried our best to ensure that the heterogeneity across all the six data sets is minimal. So After that, we performed once this DA2 and ASV generation was completed, we performed the taxonomic profiling, and it was identified that Firmicutes and Bacteriodota were the dominant bacterial phyla across the microbial communities across both the three groups. And proteobacteria, and they were the major basically contributors, Firmicutes and Bacteriodota. Along with these two, proteobacteria and actinobacteriota, they contributed smaller fractions, but all these four were consistently present across the six datasets. However, it was noticed that the control group, they consisted of like three major genera, that is Fasciolibacterium, Roseburia, and Bloschia. They were present in a higher abundance in the control group of animals, whereas in DN group, Asteretia, Shigella, and Alistipis, they were present in higher abundance, or their abundance was on a higher side. So all these things indicated that there is a certain shift in the bacteria or the type of bacterial genera which are present across the control groups and the DN groups. And these Asteretia, Shigella, Sutrella, Klebsiella, and Parasutralia, all these are proteobacteria, and they are related with dysbiosis, gut dysbiosis. They are prominently identified as those bacteria which are related with gut dysbiosis. Hmm. Another thing what I noticed was there was differential abundance across the 90 significant bacteria as identified using the analysis of compositions of microbiomes with the bias correction 2 or ANCOM BC2 analysis. Uh, so, in specifically in case of like diabetic nephropathy organisms, Pravotella and Atropobiosi related taxa and Pravotellaceae, they were present in a higher abundance, or their abundance was increased, whereas Pravotellaceae and elusive microbiome and Gimati mondaceae related taxa along with Turicibacter, they were depleted in DN groups. So, what can be the take home message from there? That this association of gut microbiota with the disease progression, was reflected very strongly in specific taxonomic shifts than in the overall diversity of these microbiota. However, when we performed alpha diversity, what is alpha diversity? Alpha diversity is refers to diversity of the microbiota across the single group. So across these very single groups, alpha diversity did not exhibit any change. So that was very surprising. And this was assessed using Shannon index, observed feature index, Fates PD, and even Pileus evenness. All these four statistical parameters, they basically revealed that this alpha diversity remained consistent across the control group as well as the disease group. The pooled Shannon diversity index for diabetic nephropathy was 6.35 on an average, whereas for control group, this average value of the Shannon diversity was 6.26. So this is very much similar across the two groups. Whereas Wilcoxon rank sum adjusted p-value was 0.65. And so overall, what we can infer from uh, this analysis is that diabetic nephropathy associated dysbiosis, they can lead to compositional restructuring over the of the microbial community rather than broad loss of richness. What it means that if in, let's say take a hypothetical example. Suppose if in in a healthy group or the control groups, say 100 microbial species are present within the community. So it is expected, it was observed that the same number of microbial species were present in the disease group as well. But what was important is that some of those species or some of those genera may have got higher expression in the disease group, or even some of those might have been depleted in the disease group as the disease progressed from the healthy controls to the diabetes to the diabetic nephropathy stage. Uh, so to identify whether there is certain change in the microbial composition across the six data sets, so it basically revealed that there is the community structure was changing, or the bacterial community underwent certain kind of succession with progression of the disease. So this was evident from after applying various kind of like statistical data sets, like statistical analysis like Bray-Curtis, Jacquard analysis, weighted unifrac and unweighted unifrac. So the Paumanova analysis also showed statistically significant group differences across all these four matrices, which I have mentioned in the previous statement, that is Bray-Curtis, Jacquard, weighted unifrac and unweighted unifrac. So however, the statistical significance, because how it is statistically significant? Because p-value was less than 0.05, which suggests that the data sets were statistically significant. However, it does not automatically mean that there is a large biological shift, or in terms of the ecological succession among the bacterial communities as the disease is progressing. So, what can be the central biological message here? That healthy and diabetic nephropathy mice, they did not differ substantially in overall alpha diversity, means the microbiota or microbial community does not undergo a significant succession with the progression of the disease. However, beta diversity and differential abundant analysis showed significant compositional differences. What does this mean? That certain bacteria are going to experience higher expression, whereas certain bacterial genera, they witness depletion in the healthy control versus the diabetes versus the diabetic nephropathy group. So data basic our data overall supported the idea of the selective microbial shift, which can be associated with the progression of diabetic nephropathy. So overall, what was our hypothesis initially? That there is a gut kidney axis is present, and that can be responsible for the progression of the, or there can be certain association between the progression of the disease and the variation in the microbial community in uh, present in the gut. So, what are the strong points which about our study is that this reanalysis provides a uniform coverage of across different data sets across the heterogeneously available data sets. So we have basically provided a uniform pipeline or a uniform workflow, so that basically establishes the methodological consistency which was otherwise missing in the previously reported studies. Another thing is this reanalysis begins from raw data sets rather than relying only on published abundance tables. So that is, we are basically, our study has been performed from the scratch. Another important point here is that multiple independent data sets, they increase the breadth of the analysis, because the number of samples, as the number of samples are increasing, and it basically establishes the statistical significance of our study. So in future there are more possibilities that that the results obtained in our study, they can be uh, applied to a more broader microbial communities or more broader data sets. So both taxonomic composition and diversity were examined. So they also facilitate, or they also support the width of or the broad, they also establish these parameters also establish the authenticity of our study. Another important point is that we perform a statistical analysis using ANCOM BC2, which addresses the compositional nature of our microbiome data. And so, uh, next Sunday, we will be basically going forward with another research paper. And in fact, every Sunday we will be we will be reading one paper and we'll be deep diving into the methods and the results presented by the authors and what can be the critical analysis which can drive the research into the domain forward. So I hope you have like this content. In case you like our content, please do not forget to subscribe our channel and also do not forget to share this video with your peers. In fact, 80% of our viewers do not subscribe to our channel, so it is a very humble request to please subscribe to our content, to please subscribe to our channel, and also please turn on the notification for the next live session. Finally, I would like to say that research becomes easier when we learn to read papers critically and just not read their conclusions or the abstracts or the methodology in isolation. So I'll see you in the next video. Till then, take care. Goodbye. Have a nice day.